So I am Katie Irwin. I'm an MD PhD student at Johns Hopkins. I'm in the lab of Phil Wong, and we are currently working to develop a novel fluid biomarker for TDP-43 dysfunction. First off, I just want to say thank you so much to Jean for the invitation to speak here today. And thank you all for joining to listen to this talk and for overall your participation and engagement with the research community. In particular, if anyone listening has been involved in studies where you kindly donate samples such as cerebrospinal fluid, I specifically want to say a big thank you to you. Especially for work like this, it truly would not at all be possible without sample donations like that. And I know specifically for CSF, that is not the most pleasant experience to donate that sample, and especially for these studies where you know, you're supposed to go in again and again and keep donating these samples. I know that takes a lot of commitment, so I just want to say a big thank you for making research like this possible. So before I go into the research itself, I want to do a quick little background on just where I'm at in my training for reference. So as I said, I'm an MD PhD student, and I'm about smack dab in the middle of it right now. So I've done the first 2 years of medical school already, and I'm about 2 years out of 4 into my PhD. And then after my PhD, I will go back to medical school to finish that up. So the whole purpose of this very long training plan is to become a physician scientist with the goal of running my own lab, but also seeing patients as a neurologist. So in that physician scientist role, my goal will be not only to focus on understanding the science behind diseases in the lab, but also understanding the human experience of these diseases and learning from my patients and taking my patient interactions and using those to inform the questions that I ask in the lab. So all of that is to say, I'm pretty early in my research training and clinical training right now, and I'm very open to any sort of advice or feedback that those of you in the genetic ALS and FTD community might have for us in the science and clinical medicine world in terms of how we can incorporate your experiences into the work that we do, because that's so important. So to get into the research, as Jean mentioned, we have a preprint on this work out, so you can read it here for some of the details that I won't go into so much today. But as I said, we in the Wong lab are working to develop a fluid biomarker that detects the loss of function of TDP-43. Can you all see my cursor, by the way?
Yes, I see it.
Okay, great. Just want to make sure before I'm gesturing all over the screen. So TDP-43 probably does not need much of an introduction to this audience. But just to summarize, cytoplasmic aggregates of this protein TDP-43 were first noted in ALS-FTD back in 2006. And the field really focused on this stage of disease for a while because these clumpings of a protein that's normally nuclear, these clumpings of it in the cytoplasm, suggests that there's something wrong, and people are focused on how these aggregates might be toxic to cells. Instead of developing a biomarker for this stage of disease, though, we're interested in the earlier step of disease where TDP-43 is actually being lost from the nucleus in the first place to get into the cytoplasm where it will then form aggregates. So we're interested in targeting this stage of disease for a biomarker because we think that it's really early stage disease that is so critically lacking a biomarker in order to really see how early these disease processes are beginning, in order to diagnose people earlier, and definitely to get treatments to people earlier. So that's why we are targeting this loss of function that happens earlier in disease for a biomarker. So I mentioned this nuclear clearance of TDP-43. TDP-43, as I said, is normally a nuclear protein. So when it is lost from the nucleus, it can't serve its function there anymore. The critical step that we're focusing on in terms of TDP-43's function. It has a lot of functions, but one of its biggest ones is at this step right here, where your genetic information is turned into RNA messages, which then get turned into proteins, which are doing most of the work in cells. So it's at this step that TDP-43 is losing a supercritical function, and that is to repress the inclusion of what are called cryptic exons. So cryptic exons are sequences that are in these RNA messages, but they're normally not included in the final message product. And that's because TDP-43 is keeping them from being included in the final product. However, when you lose TDP-43 from the nucleus, it can't serve that role anymore. So these cryptic exons do get included into the final message product, messing up that whole message, and it can't be turned into the protein that it's supposed to be turned into. So these cryptic exons, I'm sure you all have noticed, are a pretty hot topic now, whereas they weren't really recognized until 2015 when John Ling, who now runs his own lab at Hopkins, but at the time he was a grad student in the Wong lab, he discovered these cryptic exons. And since then, they've been found in end-stage disease tissues. So autopsy samples, people have noted these cryptic exons. There's also been more evidence coming out that these cryptic exons could actually have disease-driving consequences. And they're even being targeted in a clinical trial now. But although these cryptic exons have been observed in like I said, end-stage tissues, it hasn't been known actually how early in disease these cryptic exons are relevant. So we wanted to address this problem with our biomarker. So the idea behind our biomarker is that, as I mentioned, TDP-43 is normally in the nucleus, but when it's lost from the nucleus, these cryptic exons will get included into the RNA messages. So in the vast majority of these cryptic exons, they're going to get included in such a way that it just totally messes up this RNA message. So it's going to cause problems with the RNA message that basically prevent the cell from reading the message the way that it's supposed to. And the cell is able to notice that and just fully degrade this RNA message so that it doesn't even turn into protein at all. However, in some of these cryptic exons, it's only about 3% of cryptic exons. So definitely the vast minority of cryptic exons, they will get included what we call in frame. So the RNA is read in chunks of 3 molecules at a time. So 3, then 3, then 3. If this cryptic exon is a multiple of 3, it has a multiple of 3 molecules in its sequence, it can get included in this RNA message in such a way that it doesn't mess up the reading frame of the sequence that comes after it. So if that's the case, and also if the cryptic exon itself doesn't have any sequences within it that are like problem signals for the cell will allow this transcript to still get turned into protein like it normally would. But instead of having the normal protein, you have the normal protein sequence, but somewhere in there, there's this extra cryptic sequence that's not supposed to be there. So the rest of the protein will still be there, but there's just this extra bit that wouldn't be there if TDP-43 were serving its function as it should. So the idea behind our biomarker is that we can develop our own antibodies against this new sequence in the protein, and then use these antibodies to pull these proteins out of fluids from humans. So out of human samples, right now, cerebrospinal fluid or CSF, pull these cryptic proteins, we call them, out of fluid, and then we can use these as a biomarker. So not only is this just a biomarker that can be useful in disease, but it's also answering for us a critical question of how early is this mechanism depicted here relevant in disease, which, as I mentioned, hasn't been known because we've only been able to look at these cryptic exons in autopsy tissues so far. So this would be the first way for us to actually look at these in samples that we're getting from living individuals. So we had this idea back in 2015 after John initially published about the cryptic exons, but it has taken a whole lot of time and a whole lot of effort from lots of people in the lab to get all these antibodies and screen them and everything. So just a quick look into a very long process. This is a brief depiction of how this antibody generation worked, basically. So the first thing that we had to do was identify the in-frame cryptic exons, which, as I mentioned, are only about 3% of all the cryptic exons that are TDP-43 related. So that already narrows down the pool of targets that we can use for this biomarker quite a bit. So once we identified all of the ones that are in frame and will actually be turned into proteins, we then looked at the cryptic exons that are in genes that will actually be relevant to disease. So TDP-43 is a protein that's expressed all across the body, and it can regulate cryptic exons in cell types all across the body. But of course, we wouldn't be as interested for these diseases. We wouldn't be as interested in a cryptic exon that's in a gene that is only expressed in skin cells, for example, because that's probably not as relevant to ALS-FTD or other diseases like Alzheimer's disease. So how we picked our targets was from this pool of in-frame cryptic exons. We picked cryptic exons that are in genes that we know are expressed in either all sorts of tissue types all across the body, or at are expressed in the brain and spinal cord for specific relevance to these diseases that we're interested in. So then that narrowed down our pool of potential targets even more. From there, we selected 7 cryptic exon targets for antibody development. We have more in progress now, but at the time, this was our initial pool of targets that we were working with. And then, so once we identified these, we generated antibodies against these targets by immunizing mice with the cryptic peptide, so like just the sequence of the protein that is from the cryptic exon. And then you collect immune cells from the mice, fuse those with tumor cells to make basically these like immortal antibody producing cells that we can just harvest all the antibody we want from. And then a lot of people, as I mentioned, did a lot of work to screen all these antibody lines to pick the ones that actually worked well. So the ones that bound the target of interest and didn't bind to other stuff. So there was all sorts of testing that I won't go into detail about that led us to a handful of antibodies that we work with now, basically. So once we had these antibodies, we actually had to use them in some sort of test to fully develop a biomarker. So the test that we are using for our biomarker is called an ELISA, an enzyme-linked immunosorbent assay. And the platform that we're using for this ELISA is called Meso Scale Discovery, or MSD. So this is a company that specializes in producing ELISA kits that come pre-made. So a lot of people use these kits for like amyloid beta measurement in CSF, but they also offer custom kits. So basically we can get these ELISA plates from them and then make our own assay, our own test with these plates. So basically I just have tons and tons and tons of these plates. They have 96 wells on them. I end up using 16 of these wells to put just like the purified protein of interest in, which helps me calculate the concentration of protein in our CSF samples later. But anyway, this leaves 80 wells for me to measure actual samples in, and I like to measure all of my samples in duplicates just to increase confidence in the measurements. Having repeat measurements is better. So per plate, I can measure 40 samples at a time, and one test for a plate takes about a day, but I can stagger them and do a couple in a day. So it's a pretty good way to measure a good amount of samples. And right now we're working with CSF, but this type of assay can be used with all sorts of fluid samples. Okay, so how this actually works is in each of the wells on the plate, there's a little electrode at the bottom. I coat each well individually with our cryptic antibody. So that's the antibody that we developed, the novel antibody that only we have right now against the cryptic protein of interest. So the sequence, this antibody is specific for the sequence that comes from the cryptic exon that has been included due to TDP-43 loss of function. So once I coat each of these wells with a whole bunch of these cryptic antibodies, I will add the sample into the well. So each well will get a different sample. And then that spins for a while, allowing these antibodies to pull out of the fluid the protein of interest. So if the cryptic protein is there, these antibodies will bind it. And then I wash off all of the unbound material, leaving me with just a well with this antibody, which sticks to our protein of interest. After that, I have a second antibody, which binds the regular sequence of protein. So this is the sequence that would still be there like in a normal protein without the cryptic sequence. The reason we want to work with two different antibodies instead of just our new cryptic antibody is because the chances of two different antibodies both binding the same wrong target is way lower than the odds of one antibody binding the wrong target. So this technique is called a sandwich ELISA. And what this does is just increase the specificity of our assay so that we have way lower odds of actually picking up the wrong protein. And then the way that I'm actually able to detect signal from this sandwich is there's another antibody that MSD offers that will bind to the top of the sandwich antibody. And this antibody has a sulfo tag attached to it, which when you apply electricity to the plate, so the whole plate goes in this plate reader machine, which will apply electricity to the wells, generating this electrochemical reaction, which ends up producing light. And it's this light that the machine can quantify. So the light signal that the machine detects from each individual well can be turned back into a measurement of how much protein was detected in that well. So that's the gist of how this works and how I get some sort of measurement of cryptic protein from a bunch of CSF samples. So as I mentioned, we developed antibodies against several different targets. The one that we've moved forward with at this point is the cryptic exon in a protein called hepatoma-derived growth factor-like protein 2, HDGFL2, which I will just call HDG for short. The function of this protein is not particularly important in the context of this talk. It's just the fact that this protein contains a cryptic exon is what's important here. So it's not the protein itself for any nature of the protein or any function that it plays that's of interest to us. It's the fact that we can detect a cryptic exon that has been included into this protein, which is offering a functional readout of TDP-43 loss of function. So that's the important part here. We do have other targets, like I mentioned, but our antibodies against this target just worked really well right off the bat. So it was just the one at this point that we've moved forward with the farthest, but we do have other targets that we're working on. So after we identified our first cryptic antibody to start testing, this cryptic HDG antibody, we didn't go immediately into testing CSF samples. As you all know, these CSF samples are very precious. So we want to make sure that we have a well-functioning test before we start just using up a bunch of samples. So the way that we did that, the super minute details aren't that important here. But the way that we did that is we took 2 different cell groups. So one group of cells we had artificially knocked down TDP-43. So we reduced the levels of TDP-43 in these cells, which would cause them to express this cryptic HDG protein as well as other cryptic proteins, but the cryptic HDG protein is the one that we're measuring here. So this one group of cells that we have made express this cryptic HDG protein by depleting them of TDP-43, and then just wild type, normal, untreated cells. So as expected, the cells that we've knocked down TDP-43 in, which I'm calling SITDP here, these cells have higher signal on this test than the control cells, which is to be expected because these SITDP cells do have cryptic HGG in it. We know that it's there, and the normal cells don't have it in there. So we should see higher signal in these SITDP cells. And then what I'm doing here to show that our test is actually specific for this cryptic HGG protein is I'm basically messing up each of these different parts of the test and seeing if we lose signal. So if I mess up this cryptic antibody, for example, I mess up its ability to bind this protein, and I still get elevated signal in the cells that we've knocked down TDP-43 in, then that's a problem because I shouldn't be able to detect signal anymore if I messed up this antibody. That means the signal that I'm getting is coming from somewhere else. So these were the tests that I ran. I messed up this protein in such a way that it would be unable to bind to the target anymore, and we lost the elevated signal. And then I messed up this antibody in such a way that it wouldn't be able to bind the protein of interest anymore, and we lost the signal. And then I also replaced this with a totally unrelated antibody, which shouldn't bind to the protein at all, and we also lost the signal. So in short, all of these tests were just to show that, yes, our test is actually picking up what we want it to pick up. These are really important tests to do because antibodies, while they're awesome scientific tools, they can be finicky and they can pick up things that you're not expecting them to pick up. So these tests were important for us to show that, yes, this test is actually picking up what we wanted to pick up. Even though we do have this sandwich, ELISA, which I mentioned, should reduce the odds of picking up the wrong thing. We just wanted to double check and did gather evidence that the assay is working well and it is specific for this cryptic sequence in HDG. So with that encouraging data, we also wanted to test the utility of this assay in CSF. So as I have mentioned a couple of times, we know these CSF samples are quite precious. A lot of researchers want them because there's a lot of important questions to ask in the field, and everybody wants to work with these samples that are so useful. So the next thing that we checked was if this test would still work with CSF that's been thawed before. So in CSF, it has to be kept frozen so that it doesn't get the proteins in the CSF so that they don't get degraded. But every time you freeze CSF and then thaw it again, it hurts all the proteins in it a little bit. You lose some ability often to detect what you're looking for in the CSF. So a lot of people really want CSF samples that haven't been thawed before. They went straight to the freezer and never came out. But it's super beneficial if you actually can work with CSF that's been thawed before, because a lot of people won't use the full tube of CSF that they've been given. So it goes back into the freezer and then could potentially be used by someone else. So it's a way to get more bang for your buck out of the CSF that you're working with. So I'm just showing here that we tested CSF that had gone to the freezer and had never been thawed. And then we also tested CSF that had gone to the CSF, been pulled out, thawed, used, put back in the freezer, and then thawed again for our assay. And what I'm showing here is that although we do lose a little bit of absolute signal, the relationships between all the samples stay the same, suggesting that we could probably still use this test for CSF that has been thawed before. Okay, so now to the actual patient versus control group. data. We started off using this test in just a small pilot study to see if it would work at all. And very encouragingly, in the 5 CSF samples that we got from C9-linked ALS, these samples were significantly higher in cryptic HDG signal compared to our control group. So that was, of course, very encouraging for us, leading us to move into a larger study. So in this cohort, every person has a C9orf72 mutation. We had CSF from 31 people with symptomatic ALS, ALS and FTD or FTD. We also had CSF from 15 people without symptoms, but with C9 mutation. Our cohort was almost 50-50 male-female. The average age of this cohort was 52. And so this is actually a longitudinal cohort. So from some of these individuals, we have 2 or 3 or even 4 CSF samples taken at different time points. So when I say the average age of this cohort, I mean the average age of each of the people at each of the time points of CSF. So one person in this study could have CSF from age 48, 49, 50, and 51, for example. And then when looking at those with symptomatic ALS, ALS-FTD, or FTD, there was no difference between males and females in terms of cryptic HDG signal. However, when looking at the cohort overall, males did have a higher average cryptic HDG signal than females did in their CSF. But when looking at just those who were symptomatic, there was no difference. So here are our main findings from this bigger cohort that we tested. Notably, we do see elevated cryptic HDG levels in symptomatic disease compared to controls. But really, the biggest thing here for us was that even pre-symptomatically, we see higher levels, significantly higher levels of this cryptic HDG protein compared to controls. So this is telling us that this TDP-43 function of repressing cryptic exons is already compromised before people even develop clinical symptoms, suggesting that indeed this is a super early disease process that happens in at least in C9-linked ALS-FTD. And of course, the field is still working on exactly how this TDP-43 loss of function could be disease driving. But the fact, either way, if it's disease driving or not, the fact that we can pick up this disease process pre-symptomatically means that it can fill the role, hopefully, of an early biomarker that the field is so critically needed. So, and just among the symptomatic C9 carriers in this cohort, interestingly, we saw a significantly negative correlation between disease duration and these cryptic HDG levels that we measured in CSF. So this means that it appears that cryptic HDG tends to actually be higher earlier in disease. So I'm going to go back to something that I kind of breezed over a minute ago. You might have noticed that our sporadic ALS CSF did not have significantly higher levels of this cryptic HDG protein compared to controls, unlike the presymptomatic and symptomatic C9, which were significantly elevated. We still think it's too early to really make a conclusion about the sporadic ALS samples, but I want to offer a couple potential explanations for why this is not elevated. So one could be that TDP-43 dysfunction happens at a different rate, at a different timeline in sporadic ALS compared to C9. That is an option. Another is for sporadic ALS, we tend to be getting CSF samples later in disease than we're getting from C9 because of this delay in diagnosing sporadic ALS compared to the earlier recruitment into a study of people who are at known genetic risk of these diseases. And since I was saying earlier that these cryptic HDG levels tend to be higher earlier in disease, if we are capturing these people with sporadic ALS later in disease, they might have already passed this peak of high cryptic HDG levels and be well on the downtrend. So that's another option. We are currently working on testing a lot more sporadic ALS samples. It's really hard to draw major conclusions from a sample size of 6. So hopefully we'll have more data on that soon and be able to answer some more questions. But I just wanted to note that there, so I don't fully breeze by the sporadic data that we're really still trying to evaluate and answer more questions on. But as I mentioned, some of the major findings from here are that we do detect this cryptic HDG presymptomatically, and it does look like these levels actually tend to be higher earlier in disease and potentially decrease as the disease progresses. And the fact that we're seeing a dynamic change in cryptic HDG levels over time suggests that it could have even more biomarker utility versus something that you're just answering, is it there, is it not, yes or no. These dynamics are really interesting. One interesting biomarker dynamic that is being explored more in the field right now is neurofilament, as I'm sure you guys are aware. So Michael Benatar's group has done a lot of really interesting work on neurofilament, and they have suggested that neurofilament levels tend to rise early in disease, even presymptomatically, and then they reach a high level in symptomatic disease and plateau at that point. But they suggest that while neurofilament reflects the degeneration of the axons in the neurons, there could be an even earlier biomarker that reflects a different molecular process. And we think our biomarker could fill a role like this. So we propose this interesting kind of staging scale of ALS-FTD based on some of our findings so far. So as I mentioned, it's been shown that neurofilament starts to increase presymptomatically, but then plateaus in symptomatic disease. This is actually pretty different from what we've seen in cryptic HDG so far, where we think that these cryptic HDG levels rise actually even earlier presymptomatically than neurofilament. And then as disease goes onward, these levels start to drop off. I want to emphasize that this curve is not a definite, this is not something that we know right now. This trajectory is something that we're kind of hypothesizing right now based on the data that we have seen so far. But the exact shape of this is still to be determined and needs more longitudinal study. So we need to study more CSF samples from individuals at multiple time points rather than looking cross-sectionally at a bunch of different people at just one time point and trying to piece together this curve. So while we are confident that cryptic HDG is elevating presymptomatically, and then we think it's going down later in disease, the exact shape of this curve is still to be determined. But because this curve increases presymptomatically and decreases during disease, which is opposite from neurofilament, we think that using these two biomarkers in one ratio could be really effective. So we hypothesized that because of this difference early on in disease, if you take a ratio of neurofilament in CSF over cryptic HDG in CSF, we expected this ratio to actually be less than 1 presymptomatically. And then as a person transitions to symptomatic disease, we expected this ratio to become greater than 1 as neurofilament is now higher than HDG. So we were actually able to measure this in the CSF samples that we had because we were collaborating with Biogen on this project, and they had done all the neurofilament measures in these same CSF samples that we had measured for cryptic HDG. So we calculated all these ratios, and we actually did find that in almost all of the pre-symptomatic CSF samples, this ratio was less than 1 as predicted, and in almost all of the symptomatic C9 CSF samples, the ratio was greater than 1 as predicted. Something actually I think is pretty interesting to point out. You'll see that there's these two pre-symptomatic CSF samples that don't quite fit the less than 1 prediction. These 2 CSF samples are actually from the same individual, and this individual was still classified technically in their clinical diagnosis as asymptomatic, although their ALS functional rating score had actually started to decrease several points. So this person seemed to be on the cusp of reaching symptomatic disease, which actually does line up with our hypothesis of this ratio getting greater as someone starts to progress into symptomatic disease. So to conclude these findings, we find that this one cryptic target that we focused on so far, cryptic HDG, can be detected presymptomatically in C9 or 72 CSF. And this cryptic HDG protein, the fact that we can detect it in CSF means that TDP-43 is not serving its function that it's supposed to be serving in the nucleus. So we can conclude from that that TDP-43 is already being depleted from the nucleus, even before clinical symptoms can be detected in C9. And we also found that cryptic HDG levels tended to be higher earlier in disease and decrease over time. We're still working on elucidating this dynamic, but we think that nailing down what this curve looks like, what this trajectory of cryptic HDG levels looks like in disease, as well as the different trajectories of other targets that we're working on characterizing right now. We think that combining all these curves together will provide more information on disease staging, phenoconversion, and hopefully other features of disease as well. So we're pretty hopeful for these biomarkers and how they may be used in the future. We're hopeful that they will help identify disease activity before the onset of clinical symptoms, which would be so important because we tend not to treat people until we can show that their disease processes have started happening. And right now, we don't have a good way of doing that besides just waiting until someone develops symptoms. Hopefully, neurofilament is starting to play a bigger role in that as well. But if these cryptic peptide biomarkers can be used even earlier in disease to show that there are actual pathological processes going on in the cells, hopefully we can give people treatments earlier. We're also hopeful that maybe these biomarkers could be helpful for predicting when phenoconversion may occur, if that information is wanted by an individual. We're also hopeful that these biomarkers, since we think they're involved early in disease, they could help with diagnosing people earlier, especially in sporadic ALS and FTD, which have an even bigger problem with timely diagnosis. And then also we're hopeful that these biomarkers could be used in clinical trials to actually measure the efficacy of the drugs that are being tested. I think a lot of you are familiar, but a big problem that a lot of clinical trials have faced is the ability to actually measure if the treatment is doing what it's supposed to be doing. So we're hopeful that these biomarkers could potentially be used in the future to measure therapeutic efficacy for drugs that are designed to fix TDP-43 function. And we're also hopeful that these biomarkers could be used for other diseases like Alzheimer's disease, which a lot of cases do also have TDP-43 copathology. So what we're working on now towards these goals, I mentioned that we need a lot more longitudinal CSF samples, and we actually are getting a lot of those now, so we're working on generating this data. What is super helpful is CSF from people from the same individual, both before they develop symptoms and after, so we can actually map what their cryptic protein biomarker levels look like as they undergo that phenoconversion period. So thankfully, we do have a lot more longitudinal samples now. And hopefully, as we work on revising our manuscript that hopefully will be out in a journal soon, hopefully that final version will have more of this longitudinal data to give more information on this curve that I was hypothesizing earlier. We also just want to expand our sample sizes of both familial and sporadic ALS and FTD, just to get the more information, the better, of course. And as I mentioned earlier, we know that a lumbar puncture to get spinal fluid is not an enjoyable procedure. It is much easier to have blood drawn, much more pleasant. So we're working on testing this assay in blood. Still not sure yet how that'll turn out, but we're definitely doing everything we can to see if this assay is also feasible to do in blood samples. So with that, I just want to say thank you to you all for tuning in. A big thank you to all the members of the Wong Lab, especially previous members as well. As I mentioned, this work has been going on for years. So much work went into developing these antibodies and screening them, even before I was at Hopkins. So I have a lot of people to thank for the work that we're doing now, and then also a lot of awesome collaborators both at Hopkins and at other institutions, like the NIH, MassGen, and Biogen as well. And then also to our funding sources. So with that, I am happy to take any questions if there are some. One thing I would like to point out, while I mentioned earlier, I would love to hear like any feedback or anything from anyone. The one thing I ask that is not shared with me is if anyone has been involved in any particular studies. Thank you so much, but for the research integrity purposes, I cannot know if anyone has participated in any particular trials. I can't receive any sort of identifying information that might be tied to samples that I either have tested or may test in the future. So I just ask that you refrain from letting me know any particular studies that anyone has participated in.
Wonderful. Thank you so much for that, and congratulations on getting this work out there. I'm sure we have lots of interested questions and comments in from the team. I've been jotting some down, so I'll just jump into it with one question, one comment, and then I'll open it up for the rest of the folks. If people can raise their hand, that might make it easiest. So a question, so really the biggest volume of samples that exist for the C9 community are the GenFi and ALS-FTD studies. And are you collaborating with them? And one brief comment, I saw the theoretical chart that you have with the fully pre, and then the prodromal, and then the fully symptomatic crossover with the NFL and the cryptic exons. But I just want to make clear, we really don't want any symptoms. So from the patient community, we don't want the mild symptoms. We want no symptoms. Mm-hmm. So just wanted to make clear, I know in a academic sense, not thinking about these things, the mild symptoms might seem like the ideal time to treat. For us, no go. We don't want none. So just wanted to share that comment. So I'll let your reaction, and then we'll open it up for the rest of the crowd.
Yeah, no, I appreciate that comment so much. We definitely are interested in this biomarker being effective before there is any hint of symptoms. That is definitely the ideal case for this biomarker. I don't get super detailed clinical information in all cases. The most recent cohort that I've run, I'm still waiting on the ALS functional rating scale info to come in. I'm hoping when our final manuscript comes out, we might have more information about how early in disease we can pick up this marker, and hopefully we'll be able to show that we can pick this up when somebody has not complained of any hint of symptoms at all yet. So we don't have data on that to show right now, and we don't have detailed data on that. But I totally agree, and I appreciate that comment that the prodromal phase is still not early enough. Truly asymptomatic is when we're hoping we can still detect these disease processes to get treatments in there before any symptoms do occur. And then in terms of the cohorts that we're working with, the two that you mentioned, we are not yet working with right now, but there are definitely some that we are looking into.
Great, thank you so much. Appreciate your understanding of our sentiments. I want to open it up. Anyone? Dr. Edelstein, Kara, anyone, please jump in as you would. There's no hand raised, so I think you can just jump in.
Hey, Katie, thanks so much for your presentation. It was really interesting. I was really interested in what you said about not finding the same effect in the sporadic ALS samples that you had. Have you looked into any other genetic forms of ALS or FTD and besides the C9? And I'm also curious, like how, I know you have more samples that you'd want to test to see if this effect still plays out, but if we think that the biomarker kind of goes down towards the more severe symptoms of the disease, how well do you think this is going to be good to pick up, like as a biomarker?
Yeah, so in terms of it potentially going down later in disease, that is a big question that we still have for sporadic ALS. We don't know yet if these levels, because as I showed, we've only tested 6 samples at this point, which is not a whole lot of information to rely on for us now. So we need to test a lot more samples. We have some on the way soon that will hopefully give us more information, but we don't yet know if the cryptic protein levels are going to look the same between genetic and sporadic. And we don't know yet how early this will be relevant in sporadic, because people who are going to develop sporadic ALS, especially if this is still a CSF study and not something in the blood that's right now, in blood at the moment, people aren't going to be coming in to get their CSF drawn to test if they might develop sporadic ALS. So the clinical utility of this biomarker for sporadic disease will definitely depend on what we see when we figure out these dynamics a little bit more, because if we aren't able to detect the protein later in disease when these patients are actually being identified, yeah, you're totally right, it might not be as relevant for them. But we're hoping we will still see elevations of this cryptic protein at a time point where people might start to notice that something is off and come into the clinic for diagnosis. And then we could hopefully get them diagnosed with ALS earlier than they would have been otherwise and on treatment earlier. In terms of the other genetic causes that you were mentioning, we do have some SOD1 samples that I'm in the process of analyzing right now. I'm not sure yet if that data will make it into our paper that will hopefully come out in a journal soon. But we are working on that, and hopefully either way, that data will be available soon. But yeah, we're interested in other genetic causes as well.
We're approaching the hour. I don't want to keep anyone too late. Does anyone else have a question or a comment? I did want to just jump in on some things, not hearing others. So has your lab been considering the work from GenFi All-FTD showing the brain volume difference in the 20s for C9 carriers? And, you know, that's well documented at this point, been almost 10 years, or, 8 years that's been in the literature. So just curious.
Yeah, we haven't looked at specifics of that data, but we have definitely taken in the overall conclusion that a lot of studies have put out there, that there are noticeable, detectable differences in different capacities. Brain volume is one, for example, that are detectable super, super early, which is why it was interesting but not surprising for us that we were able to detect the TDP-43 dysfunction early on. We haven't, I don't think we have tested samples from that early in disease yet. But like I mentioned, we're still trying to pin down exactly how early these cryptic proteins can be detected, and definitely are keeping in mind that there are noticeable differences between C9 carriers and control individuals in a lot of different aspects. And I know the field is still trying to pin down exactly what initiating pathogenic mechanisms might be.
Wonderful. Thank you for that. Also in that vein, there's some literature on surgical tissues from people with C9 FTD. Are you familiar with what I'm talking about? And they did not find TDP-43 aggregates. and but they already had FTD symptoms. And the papers were, guessing that, oh, maybe TDP-43 is not part of it, but maybe they would find these exons in those tissues. So I don't know if this might be looked into.
Yeah, I am most familiar with one specific case where a woman was having resection of part of her temporal lobe for epilepsy treatment, and then ended up developing symptoms of FTD years later. So they went back and looked at the brain tissue, and they saw that there weren't any cytoplasmic aggregates yet, but there was TDP-43 nuclear clearance. And these cryptic antibodies that we have now weren't available at the time, so they weren't able to look for the presence of these cryptic exons. But it's cases like these that have shown the nuclear clearance without cytoplasmic aggregates that provide a lot of backing for the work that we're doing. And I think we have a lot of resources in our lab that if we were able to get surgical tissues, we could do some interesting studies with those.
I want to tell you, we are very interested in setting up a process by, if people in our community know that they're going to be going in for voluntary surgeries, that they could be donating those things or working with people to do. So anyway, if you're in conversations where people are talking about that, very happy to connect and figure stuff out.
Yeah, that's awesome. Thank you. Yeah,
and there is.
Oh, sorry. Go ahead.
Sorry. Well, yeah. Let's. Happy to be in touch.
Okay. Yeah.
There is one more question, and we're almost at the hour. But in the chat, is this information being shared with our local ALS clinics? In other words, when do you think the doctors and geneticists will have this information and be able to talk to people about it?
Yeah. That's a great question. Thank you. I wish science were so much faster than it is. I imagine there's going to be, I don't know all the details being kind of early in this field, but I imagine there's going to be quite a few steps to get this biomarker actually into the clinic. We're, of course, working on our end, as hard as we can to get this data out there as quick as possible to start moving forward with some of these things where it can actually be utilized with clinicians and the patients that they care for. But in terms of the exact timeline of that, I don't know the specifics of how that will play out. But we're hoping for as fast as possible.
Well, thank you very much. It's a wonderful note to end on. I do want to ask you, I have some more things here. Could I email you some further stuff and maybe open it up to the rest of the people? If they think of something later, they could email me and then I could get it to you.
Yeah, of course. Happy to chat.
One person raised their hand, or maybe they took it down. Okay. Well, thank you so much. And we really appreciate it.
Thank you for having me.
Hope everyone has a great day.